welcome back to VLI Bio Labs Online. My name is Ms. Copay, and today we're doing Lab 6, our final one for the semester. And this is going to be a combination actually of two labs in your lab manual. So we're going to cover how to construct our DNA model, and then we're going to be looking at protein synthesis and mutations in DNA and how they affect our proteins. So today in your kit, really what you're going to need is your pipe cleaners. This is going to create our double strands of DNA as the backbones of our molecule. And then we're going to use toothpicks. You might need to supplement with additional toothpicks, but those are there. Now what you do need to get from the store probably is some red and black licorice. And you want to make sure that it's the red vines because they're hollow in the center, and we're going to see how we're going to string them actually onto our pipe cleaners to create our backbone. And we need red and black because if you remember, the backbone of DNA is a sugar phosphate backbone. So one of these is going to represent our sugar and the other is going to represent our phosphate that alternate. And then we have our gumdrops, and you can see there's 4 different colors, and hopefully that automatically makes you think of the different 4 nitrogen bases. We could make that A, T, C, and G. So we're going to be constructing our nucleotides. And I'm going to walk you through this. It's a short procedure to get started, but then you're going to make sure you follow your lab manual. Remember that I said in your lab manual you're going to be doing, the first one is the DNA model building. That's lab 9, starting on page 53. And in here is going to have you drawing and explaining your model as you go through here. And then you're going to jump forward to lab 11, which starts on page 71. And that is going to be looking at protein synthesis activities for how our genetic code, the universal code for building our amino acids and protein synthesis, comes from our DNA code. And so we'll be looking at mutations in that component too. So today what I want to cover with you in our introduction here is how to put together your DNA model. And so in here we're going to take a knife or scissors, and the key is to cut your red and black licorice into substantial size so that it holds its hollow shape in the center. If you cut it too small, it will fall apart. So let's talk about what one nucleotide is. So if you think about one nucleotide, I'm going to cut these just like this. Hopefully that's not going to. I can use my scissors or I could use a knife. Okay, so one nucleotide would consist of a sugar bound to a phosphate group. Okay, and then we would have our nitrogen containing base attached to the sugar. And so if I put a toothpick here, I'm going to use a toothpick here to anchor it to my sugar backbone, and then I would have a phosphate. And that would be one single nucleotide. And in this case, if this was my T, this would be my thymine nucleotide. So we're going to first start by building our backbone. So I just wanted to make sure you recognize that even though I'm going to build my model one way, the actual way a DNA model is constructed is by having all of the nucleotides laid out, and then they attach to one another in the correct order. So the way we're going to construct them is actually not the way our body would construct a model of DNA. So your lab manual has you take sugar here. So we're going to take our red for our sugar and feed him through. And then I'm going to take a phosphate group, and I'm going to make my backbone here. I'm going to slide him down, hoping he stays in place, okay. And then I'm going to alternate with my sugar and phosphates. So here's my next sugar, and these are attached covalently through phosphodiester bonds. Okay, and then my phosphate. All right, so I'm going to repeat this. Oh, so be careful with your licorice that it doesn't start falling apart on you. So I would have 2 of these backbones constructed, one going in one direction and the other going anti-parallel to it in the opposite direction. Okay, so there's our sugar phosphate backbone. Now, attached to each sugar is a nitrogen-containing base. And so I'm going to use my toothpick here, and I'm going to shove it through and attach it to the sugar. And then depending on which nitrogen base I'm looking at, if my nucleotide was a T, I would have a T on this side. Okay, so here would be my thymine. Right? And then I could have another nucleotide here attached to that sugar, and we could make it, let's say it's a guanine here. Okay. And so we could record these on our tray here by labeling them in certain colors. And if this is an adenine, I can put another adenine. So now I have a single strand of nucleic acids representing 3 nucleotides. So here, if I read this based on their identity, I would say this is adenine in green, guanine in orange, and thymine in purple. And that would be 1 strand. Now I can construct the opposite strand here, doing the same procedure. And so I can take another 2 strands and make the complementary base. And remember here that my thymine is going to pair with adenine. So our base pairing rule applies here, so I would construct this. And they're held together by a hydrogen bond. And so our DNA molecule actually is how our 2 strands of the DNA molecule come together, is through that hydrogen bonding of our nitrogen-containing bases that make up the middle of our ladder. And so if I push him on here, I can make my nitrogen bases represented to bring my sides of the ladder together. And so you're going to have to construct both sides of your DNA molecule using this procedure here until you have a nice double helix. So now we're going to build the opposite strand. Remember that they don't run parallel. So if I had sugar, phosphate, sugar, phosphate, sugar, phosphate, my leading end of the other one would actually be going the opposite direction. So my free end would not be a 5' phosphate group up here. This leading end on the 5' phosphate would be headed in the other direction. And so we will actually build our second strand anti-parallel using our repeating sugar phosphate going in the opposite direction. So you should end up with the free phosphate, the 5' free phosphate on opposite ends to the 3' hydroxyl sugar on your DNA molecule. Okay? So there's my final DNA model. And remember, this would be a helix, so it would actually start to turn on itself. If you had 5 base pairs, it would start to make actually a nice turn. So there you go. Keep working on it. It's going to look great. You'll be so proud, and then you'll get to say, hey, this is your DNA. So here's your final DNA product, and go ahead and show everybody your little twisted helix and say, this is my DNA. Bye. So that's what you're doing in lab today. And you're going to answer the questions in your lab manual and turn in both of those lab reports from lab number 9 and lab number 11 in your lab manual, along with your template for the lab report write-up for this week. Take pictures of your work so we know you did it, and let us know if you have any questions. See you next semester.